Oh, yeah. All right. So today we are going to dive back into the ocean and talk more about fish as we think about the early evolution of tetrapods. They're going to be things with four limbs, mostly living on land. I say mostly because things like whales are going to show us that some of these tetrapods eventually return to the ocean. The tetrapods are going to start out on land. We're figuring out how we get vertebrates on our land instead of just insect arthropods, like we saw living among the early plant life. So we are mostly going to be centering ourselves around the Devonian. So we're in the Paleozoic. We've been kind of talking about the Ordovician, the Silurian, and now the Devonian, bless you, all kind of together a little bit here. Okay, so we are now up to around 400 million years ago. So 419 to 359 million years ago. So in terms of what Earth looks like at this point, we don't have huge differences from where we were in the Ordovician and the Silurian. So we still have Gondwana here at the bottom in the southern hemisphere. But one thing that is happening in the early Devonian is that Europe and the Americas kind of crash together. So we have a new large continent kind of forming here. So we're starting to get kind of like two separations here. So here's this Euramerica or Eurasia or something it's also called. One of the modern day features that still exists kind of from this time period are the Appalachian Mountains. So these rose up essentially when this collision happened. So the Appalachians, part of the reason that they are shorter and kind of softer than like the Rocky Mountains is the weathering that has happened and this kind of age here of the Appalachians. In terms of the climate, because we haven't looked at any climate data in a little bit, right? So here we're looking at our CO2 concentrations here in red, the percent of atmospheric oxygen in blue. So we have two different y-axes here so that they can be charted together, as well as the temperature data put down here on the bottom for us. So this is looking from the Cambrian through the Paleozoic and then continuing actually up mostly to the present day. So this figure gets us farther than we're going right now. So if we look, let's first look at that carbon dioxide level. So notice that the carbon dioxide level during this whole time period we've been talking about is really high. So for reference, modern day CO2 levels are about 420 parts per million. And you can see here in the Cambrian, we're above 6,000 parts per million. So the atmosphere during this time period is really different. It's falling over the course of the Paleozoic, but nowhere near as low as we have today. We also have changes in atmospheric concentration of oxygen, but this is a little more in our normal range here. So we're ending up around 21% in the modern day. We can see that we have kind of lower oxygen in the Cambrian and the Ordovician. But as we start to move towards the end of the Paleozoic, we have those oxygen levels rise. As we move forward on Wednesday, we'll probably talk about it. We're also going to see that the land plants are really expanding during this time period. So temperatures are varying. So you can see that we have a dip here at the end of the Ordovician. We haven't really talked about it, but the end of the Ordovician does also correspond to our first real observable mass extinction. So people talk about essentially 5 mass extinctions in the history of life on Earth that we can directly observe anyway. So one of those is happening at the end of the Ordovician. We didn't focus on it as we were talking about the Ordovician because actually we see a lot of those same organisms that were prevalent before that mass extinction. Their diversity dies off, but a lot of those things kind of stay around and then kind of peak back up later in the Paleozoic. So we don't have a huge turnover in the types of organisms we see, although we have a big die off in terms of like genera and species. When we have that dip in temperature, getting really cold, we have some glaciation happening, so that's kind of corresponding to that first mass extinction. Yeah. Now that we're into the Silurian and the Devonian, we can see that it's starting to get hotter. So hotter than the present day. Let's say let's see this graph. So around 25 degrees Celsius as our best average from a bunch of reconstructions of the data. So these represent different papers that have estimated what the temperature would be. So you can see that we have at least one outlier on this data, Song et al. In 2019. Thinks it was really, really hot in the Cambrian. You know, they have slightly different interpretations there, but otherwise these other papers are all getting pretty similar numbers based on that. So as we're going into the Silurian and the Devonian, these carbon dioxide levels are starting to go down, oxygen levels are starting to rise up, and we've got a nice kind of warm Earth at this point. We're going back into the ocean for a little bit. So in the mid-Devonian, a reef might look something like this. So we have the evolution of corals throughout this time period that we've been talking about. So now we would have an actual kind of coral reef structure. Corals are animals kind of like living as little collectives. And we'll have some cephalopods, some trilobites, some crinoids, so just other organisms that were present, as well as the brachiopods, those two-shelled organisms that we were looking at that were not our bivalves. We have a good index fossil for the Devonian. So ammonoid or ammonite are these swirling kind of structures. So they look a little bit like a snail. You've probably seen one like sliced in half and polished at some point. You can wander around the room, see if there is one. Here, there probably is. But these are present and very common from the Devonian through the Cretaceous. Even though they look like snails, they aren't snails. They do die off. They do go extinct. So that can really put us to the correct time period. They have a distinct kind of suture pattern, so a distinct point at which the shells kind of come together. And they're really widespread, which is very helpful. So during the mid-Devonian, in the oceans, we're starting to see these extensive reef complexes forming. So lots of diversity in these reefs and kind of big structures and big communities. One cool group that we have not talked about are the Eurypteridae or the Eurypterids. Okay, so these are giant sea scorpions. There we go, there's that giant sea scorpion. Of course, this one looks a little kind of trilobite-y, but our Eurypterids are referred to as giant scorpions, and when we say giant, we mean like 8 feet long. So this group includes the largest ever arthropod. Arthropods being those segmented animals with their paired jointed limbs. The record for them goes back to the Ordovician, but they're really sparse back then. And we really start to see them kind of like everywhere in the middle Silurian into the early Devonian. So if you were swimming around in the ocean at this time period, I would be frightened. So here's an average human compared to the size on some of our Eurypterids. You can see we've got some truly enormous creatures here. While we are in the ocean, we're going to be thinking about fishes. So the Devonian is often referred to as the age of fishes because of the massive diversification that we see. So we talked a little bit, started out thinking about fish as we were thinking about practicing some of our phylogenetic terms and looking at some of our early jawless fish. But now we're really going to kind of go through the whole kind of tree of fish as we move forward to the evolution of tetrapods. So pull the controls so they're not covering. All right. So we can see in the Cambrian, our vertebrates that we're starting out with are jawless. And then we start to have this diversification starting in the Ordovician, moving into the Silurian. You can see here in the Silurian and then in the Devonian, we have lots and lots of different types of fish present. So when we were talking before, we were looking at our jawless fish, the echinathans. So we were starting. So we saw one group, the Strachoderms, we kind of focused in on with their protected heads. Those are the skulls that were protecting them. And moving into the jawed vertebrates, we took a little look at the Placoderms that kind of retain some of those traits. Okay. So jawless fish look like this. The only real jawless fish that we see still present in the modern day are our hagfish and lamprey. So we can see that those are breaking off from the rest of this fish tree quite early. So we can see that they're almost an outgroup kind of here, but they're worth looking at because they are so creepy. So here's a picture of, I believe, a lamprey on bottom and a hagfish on the top. So not having jaws definitely does not mean that they can't eat stuff. So we can see these awesome rows of teeth here in our modern day echinathans. So they're pretty gnarly looking, but they're not really fish shaped. So you can see how they're kind of weird compared to what you think of as a fish. Almost more like an eel, but they almost look like they don't have a head. The two have a head because they have a front end, but we don't see that sort of like neck structure. Eels have jaws, these don't have jaws. They're real weird compared to our modern groups. So when we're in the Devonian, our extinct groups of jawless fishes, some of them look a bit more normal and a bit more fish-like, which may have to do partially with the fact that those hagfish and lamprey were a really early branch off of this fish tree up here. Whereas we're moving a little bit more towards the jawed fishes when we look at a bunch of the extinct groups. So they would have had a more recent common ancestor with our existing jawed fishes, even though they're dead now. So our echinathans and those Strachoderms are really thriving in the early Devonian, but they're going to start to kind of die off as we move forward in time. So into the middle Devonian and the late Devonian, that's when we're going to see the jawed fish diversifying and rising in terms of how prevalent they are in the environment. So we're going to see different types of jawed fish, and we're going to see more jawed fish overall as they become much more efficient predators. They also become more efficient herbivores. They're going to be using those jaws to eat things. So the fish that we see with jaws in this time period are going to be the acanthodians and the predators. So we're looking back at this tree to think about those jawed fish. So our jawless vertebrates are up here. So we still have hagfish and lamprey, So we can see how they're that really early branch, whereas some of the other jawless fishes that we looked at, particularly those with stracoderms, have much more common ancestry with our jawed vertebrates. So as we're moving into the later parts of this time period, we're starting to see all those splits among the jawed vertebrates, which are also known as gnathostomes. So the gnathostomes have jaws, eventually going to be able to do stuff like bite and chew. We can also see this sort of close association here between those on this chart, osteostracans, so like our ostracoderms and the placoderms. So there's some debate over the exact sort of lineage here, that relationship between the two, but they're both kind of armored. They're very similar looking kind of fish, but we do start to see that development of the jaw structure in the placoderms. So while we expect that they had a very close common ancestor, interpret the placoderms as branching off a little bit further because they have that synapomorphy with our jawed vertebrates. So we can also see the acanthodians here. They are also extinct, but they are a sister group to the chondrichthyes, which are a modern group of fish, including sharks. Let's think for a second about the evolution of jaws. Okay. So while pretty clear to us why jaws are helpful for us, this is like chewing and feeding is not actually why we think that early jaws evolved, which takes some thinking about development. So if we're looking at a jawless fish, we're going to see a skull up top, might be made of cartilage. And then we see gill arches and gill slits down under that skull. And this is actually also true in even early human development. Well, embryos essentially have these gill structures that eventually become all sorts of different parts of your head and neck. So we have gill arches and gill slits. So we think that the early jawed fish are taking those first steps of gill arches here. And that first set of gill arches is becoming this jaw structure. So in our early jawed fish, we actually don't even see teeth. All right, so not biting really, although I guess you can bite something even if you don't have teeth. But we think what they were actually doing was helping them breathe. So you think about a jaw, we eat with it, but it allows you to open and close your mouth, which means that it can also kind of help you pump air almost. So we think that these early jaws were actually helping them sort of push a lot more air kind of over their respiratory structures. And they've been interpreted as breathing through a pumping action, kind of actually like modern day frogs. So modern day frogs do this kind of like buckle breathing. So we think that's kind of what was going on with these early jawed fishes. The modern jawed fishes, including the cartilaginous fishes like sharks and rayfin fishes, which are going to look more like your typical fish, have added on additional gill arches to this early jaw structure, turned them into bone, and added teeth, and sort of modified this early jaw structure into a feeding focus structure, which brings us to a vocabulary term here. So exaptation refers to a process or like a feature that evolved for a different reason, right? But the initial selective advantage is not the modern use of that same feature. So this early jaw structure we think was associated with breathing, but this modern structure is really associated with eating. So we see this kind of change in the functionality of the structure. So looking a bit more at this evolution of the jaw. So here again, we can see those gill slits and the skull. And then we can see some of these gills looking like skeletal rods. So we've highlighted two of them, right? So they become kind of skeletonized. And then we can see them moving forward towards the mouth. Right? So we can see them moving forward and then becoming incorporated in this jaw structure. Slightly familiar friends, the Hox genes, as well as some other related genes, control large developmental patterns, including these jaw structures. So here we can see some of the associated Hox genes that are associated with these different branchial arches, so these different gill arches. So this is something we can actually look at, see where these genes are expressed in both our existing jawless fishes as well as our existing jawed fishes. So we can see that relationship even in the modern day. It also happens to be the case that in your early gill arches that you have during embryonic and fetal development also do become bone structures in your skull, like they become like bones in your ear, bones in your neck. So this idea of going from like a gill structure to a bone structure may seem crazy, but it actually not all that uncommon. Things that we can actually kind of see develop in real time. Taking us to a paper. Cool. All right. So just zooming in on the placoderms again. So these armored fish in the Devonian, looking a lot like our ostracoderms, but they're becoming jawed here. A real dominant predator. So again, they can be very, very large. So they're up to 20 feet long. So basically, everything in the past is way bigger than you think it might be just looking at a picture. So part of why we were looking at climate data is we can start to think about why that might be. So the different sort of prevalence of gases and climate, I think maybe has some effects on like our size constraints in earlier periods. We'll take a look at that in a while. Okay, but here we're looking at Dunkelostia, so very large placoderm. So it has a head shield, kind of like our ostracoderms did, or at least some of them. Got some bony plates in front. We got a strong jaw that has formed here. And we have some gaps and hinges, and then some more plates. So here we see a thoracic shield, but it's not totally covered in these plates. So we can see that it has this naked body as we go farther back. And here we can see one actually right here, which is crazy. I would be terrified if I saw one of those. Fish are scary. Okay, so here's you compared to some placoderms. So these are real kind of apex predators here in the Silurian and into the Devonian. You can see lots of different kinds of placoderms. Our other group that we're thinking about as our early jawed fish are the Acanthodian. So those are the ones that are more related kind of to sharks. So when we think about the development of our cartilaginous fishes, which is what our sharks are a great example of, those are called chondrichthyans. So chondrichthyes is maybe, in terms of its phylogeny, kind of nested within this group. So the acanthodians that we see in the Devonian themselves are extinct, but whether the sharks branched off within this group or whether they're sister groups is something that could be up to debate depending on the data you look at. These acanthodians definitely come first, though. They predate the sharks in our fossil record by about 50 million years. So here we can see some relationships here between our groups. So here we're looking at our invertebrates. We're focusing in on our fishes. So we see jawless vertebrates, those agnathans separating out. We have our lampreys as an example. Now we're having our jawed vertebrates separate here. We're going to see in the modern day that we really separate jawed fish into the chondrichthyes, the cartilaginous fishes, and the osteichthyes, the bony fishes. So cartilaginous fishes are mostly made of cartilage, so they will have some skeletal structures, but when you're thinking about vertebrates, you don't necessarily always have to have bone itself. It's more about the developmental structure rather than the like mineralization of those structures. And in fact, bone in development, in your development, actually starts with a cartilage model. So like, yeah, all your bones basically start as cartilage that then progressively kind of get filled in and hardened with bone. So cartilage and bone are really like tied together as types of structures. So our cartilaginous fishes are like sharks, rays, and skates. Skeletons primarily made of cartilage. We'll look at a little shark just to kind of see that structure. So we've got a skull up at the front. We've got a mouth with a jaw. We've got some gills. We got the heart here. And there we see those vertebrae running down the back and some organs here. We got some fins as well, but most of this is going to be made of cartilage. So again, another version of the phylogeny, this one a bit simpler. We're starting to zoom in on some things. So we expect that we had a jawed ancestor that was common to the placoderms, and also common to those acanthodians and the chondrichthyes, the cartilaginous fish, as well as to our bony fish. And we think that the placoderms separated off first, and before we had a common ancestor to these two sister groups. Okay. So the chondrichthyes and our bony fish are going to have a more recent common ancestor. So we're not going all the way back to the first jawed ancestor. Thinking about those bony fish, they have a bony skeleton instead of cartilage. Right? So osteichthyes, you can see over here. So we can see them continuing on to the present day. Right? So anywhere on this chart, by the way, that you see this kind of like double line, I think that's showing you that there's like a long time period continuing. Those double lined ones are the groups that we still have around today. So the rest of these where we have endpoints is really where they go extinct. So when we're thinking about the osteichthyes, so their sister groups are cartilaginous fish, so sister groups are the chondrichthyes, but within them, they're then splitting into two sister groups now, the Sarcopteridae, the lobe-fin fishes, and the Actinopteridae, the ray-fin fishes. So these two are going to look different in structure, but they're both going to have bones. So when we're looking at what we would see inside each of them, so a ray-fin fish is going to have, it's going to have like a line of symmetry down the center here, and it's like lateral to lateral, so like side to side, and it's going to swim kind of side to side. You're going to see it bend side to side, all its muscles are really going to be in the body of the fish. And so it uses those fins to swim a little bit and to sort of move water over the gills, but there aren't really muscles in these fins in our ray fin fishes. So they have just a couple like kind of bones at the base, and then these bones continuing into the fin without muscles attached to them. This is different than what we see in the lobe fin fishes, because in the lobe fin fishes, we have basically one single connector, this kind of narrowing point as we enter the fin. But then we have more bones inside the fin in a specific kind of series, a specific kind of arrangement, kind of in rows. So we can see in the ray fin fish, we just have this long little row of tiny bones. But here in the lobe fin fish, we start to see this kind of separation and this different sort of structure here. You would pass around the skeleton and take a look. There it is. Yeah. Let's take a look at the fish in our fish tank if we've got one and figure out whether we got a ray fin fish or a lobe. I can see the fins from that far away. Hey, maybe we got this somewhere. There's a bit of I don't know if this affected this fish specifically, but that corner of the room that came in with snow and stuff. I think maybe a pipe burst or something, so it kind of destroyed everything that's in that corner room. That's why it. I think there were creatures in those too. Oh no. Yeah. So what kind of fish do we have here? What are we looking at on the skeleton? The ray fin fish or a lobe fin fish? Or if you have a vote about our fish in front here, what do we got? They are both ray fin fishes, which are really our common type of fish that we see when you're thinking about bony fishes and ray fin. So we do have lobe fin fishes, but they're weirdos. So we can see on this lobe fin fish, in addition to this changing kind of bone structure in the fin, you can see this kind of like flattening of the head, right? Now it's like it's weirdly kind of smiling instead of that typical fish face up here. And so they look a little different. So the lobe fin fishes are in a different group, the Sarcopteridae. And we will zoom in on some lobe fin fishes because this is what's leading us to the development of our arthropods. So here we're looking at a lungfish. So you can see how like this is a fish, but it looks different, right? So we can see that flattening of the head. And now we can see, and realize that this fin is a fin, but it doesn't really look the same as your typical fin. So that is those muscular fins with those bones in them starting to kind of pinch off and look really different. Some of these lobefin fishes can even kind of like walk a little bit along the ocean floor, like along these fins that they're using. So this is an example of a lungfish, right? And it is also called a lungfish because it has lungs. It lives underwater, but bless you, we start to see these lung structures in parallel along either side of the notochord. So this is where we see like our spine and our spinal cord vertebrates. So here we see a left lung, and we're looking at a liver here. And this lung in our lobefin fish, and this lungfish is homologous with the lung in tetrapods. So what we're saying is it has common ancestry. It is kind of like the same structure. So this isn't an example of convergent evolution. This is an example of the same structure, looking kind of different, but based on a common ancestor. So we think the way that these lungs started to form is from a branching off of the gut tube. So like when you eat or breathe, right, food goes in your mouth, air goes in either your mouth or your nose. It starts out in this common structure in your neck called the pharynx, which then splits in you into a tube, your trachea, that goes into your lungs, so carrying the air there. And then the rest of the food, drink, whatever, continues down your esophagus into all your organs, right? So into your stomach, your large intestine, your small intestine, before coming out, rectum and the anus. Okay, so we think that what happens here, so we started with like this one common gut tube. Then we start to have this budding off. So here you can see a respiratory diverticulum. So diverticulum is just like a lump that comes off. So like a sort of diversion, a diverted kind of canal here. So we can see that pinching off of the gut and then starting to kind of close over and separate. You can see here the developing lung buds along the way. Okay. So comparing this to some fish structures. So here we have some examples of existing fish anatomy. So the tube, here's just our gut tube in each. And then on the bottom, we see the anatomy that we would expect in like a lungfish. So underneath we see the lung in this branched off whole section. Right? But in other fish, they do also have additional structures that come off of that gut tube, specifically like swim bladders. So they kind of help give fish buoyancy, help them kind of like float and control where they are in the water column. There's also a budding off of that gut tube. So we have groups of fish that have done different kind of modifications to this single digestive tract structure. So we're starting with our lobefin fishes. We got these bones and muscle in their fins here. Right? We see one lineage of these lobefin fishes that progressively get longer and longer fins, becoming more and more limb-like. This is when we start to see fish that can kind of like start to almost walk here. And these much more strongly developing fins. So here we see an example of Eusthenopteron here. This is a fossil that we see during this time period. We can see those structures kind of getting longer. Right. And this leads us to the origin of tetrapods. So I want you to take a second and kind of look at this diagram and think about what's going on. So we said that lungs in the lobefin fishes and in tetrapods are homologous, so that's true. now that we're looking at the fins, what are some structures here that would be homologous between our example Sarcopterygian and our mouse here? So how are they indicating homology on this diagram?
What color of the similar structure?
Yeah. So those colored structures are showing you the homology here. So we can see in red in our lobefin fishes, we start out with like a single clunky looking bone, but like a single bone connecting us to the body of the fish.
No,
Oh, just kind of looking. Yeah. So if you look at your own arm, because we're sitting, right? This is how your limbs are arranged too. So you have a single bone connecting your shoulder to your elbow as you start to go into the limb. Same thing is happening in your leg and your lower limb as well. Then after that single bone, we have a set of paired bones. So this is also true in your own forearm. You can kind of feel two bones there. Same thing is true in your lower leg as well. So we see the same arrangement. And then in the wrist and hand, we have a collection of bones that look somewhat similar to these additional rays from our lobefin fishes, but this is very reduced and that homology kind of isn't quite as present as we think about those rays. So it's really about these connecting bones or connecting to the body. So for thinking about the arm, like the humerus would be analogous to that first kind of structure here. And then we have our ulna and radius homologous to these two second structures. And then distal elements, distal just means like distant from the body. So like your fingers are kind of homologous to those rays in the fin. And here we have a mouse hand and a fish hand where we have, or a fish fin, sorry, not a fish hand, where we've marked the same Hox genes in each. So you can see that that's lighting up green in the same kind of region here. So when we say something is homologous, just touching base, to be homologous, something has to be genetically heritable. You have to be able to inherit it. Right? So we're talking about a similar structure that is inherited. And we want to know that that inherited similar structure is inherited from a common ancestor. So that's often when we'll look back at the fossil record to see whether we can see evidence for that trait having a common ancestor and being present in that common ancestor. We could imagine heritable traits that are present in two different groups, but arose separately evolutionarily, in which case that would be homoplasy or convergent evolution rather than homology. So here is another example of our development of lobefin fishes towards our tetrapods. So this is Acanthostega. Acanthostega has these developing limb structures. It's still fully aquatic, so it's still underwater, but you can see that it is starting to look very limb-like down here. Now, they wouldn't have been strong enough to hold this fish, we're going to call it fish, up on dry land, but it is kind of sufficient for potentially dragging it along, helping it walk along kind of the sea floor down here. In order to move to land, we actually have to kind of develop like a bony shoulder and pelvic girdle to really kind of brace ourselves to the vertebral column. So next, we've got our Ichthyostega. So here we can see that we've retained some aquatic characteristics. So the tail still has some like fin rays in it. The gills have bones that cover them over, right, which is an aquatic characteristic. But we can see that it's kind of an offshoot of the tetrapod phylogeny, and we can see that it has these developed limb structures, but now we can also see that we have a pelvis here. We can see that pelvis developing, which really gives us information about that bracing to the body. So we think that probably as we were going towards the origin of tetrapods, probably had some creatures that were sometimes kind of in the muddy tidal zone moving around, but also aquatic, so also spending some of their lifespan in water. So we don't think it was, just a sudden light switch from one to the other. Makes sense as you think about it. So we can think of them potentially being in little tide pools or like little inland lakes kind of starting to dry up. We can imagine that some of these traits would have been advantageous in these kind of changeable environmental conditions. So here we see a comparison of some of these skeletons of some of our early fish, we're saying that kind of in quotation marks, that are related to tetrapods. So exactly where we want to draw the line and no longer call something a fish. You can kind of decide where you want to draw the line, because we saw that tetrapods and land animals, including ourselves, are kind of nested within that fish taxonomy, right? So we were considering that like a separate group, but like we're within this relationship to fish. Okay, so here we see the Ichthyostega with its really strong limbs. You can see Acanthostega kind of pinched out fins. Now we're going to think about Tiktaalik, which is super cool. Our most famous example of it, one of our first creatures on land, we really think. So Tiktaalik had fins with fin ray bones, had scales, had gills like most fish, but it also had really sturdy wrist bones and neck structure. If you're thinking about like being underwater, you don't really need a neck, right? Like you don't need to like lift your head if you're swimming back and forth. So this development of a neck to be able to support and raise the head against gravity is actually a development to life on land. We got shoulders, we got thick ribs. So these are structures that really indicate to us that it's kind of quite related to our four-legged vertebrates. So you can see it reconstructed as kind of looking like the front half of a crocodile, right? But the back half, we still have some fishy looking stuff going on. All right. We're gonna watch Neil Shubin discuss finding this fossil. Finding it's pretty cool. And we're gonna speed this up a little bit.
University of Chicago, and I'm going to talk today about finding the color. My quest began in graduate my second year when I saw this section. This is the slide my professor showed back in 1986. They showed what we knew at the time the fish tetrapod transition from like water, like on the cartoon of these fossil creatures. The one on top, the fish, about 390 million years ago. The one on the bottom is a tetrapod, about 305 million. I remember the student seeing this diagram the first time thinking, holy cow, what at first, science is impossible. How to think about the water way? It seems so utterly impossible. So the thing on top is the thing on the bottom. And that's what became my class. So like any good paleontologist, I applied the paleontological tools to try to find the unique, they want for a creature to do. So how do you have to find two places? Three things. This box is the right age. The box is the right age and whatever the book interests you. So in this case, we really want something between 390 and 365 at the time period known as the late development. The other thing that's important is to have rocks of the right type. Not every kind of rock holds fossils. Some are superheated, some are highly pressurized. So we need a particular kind of rock, sedimentary rock. And not only that, sedimentary rock in the right environment. The other area, rocks of the right age, rocks of the right type. Well, it does me no good if my wonderful rocks of the right age and the right type are buried 5 miles underground. And these things have to be exposed to the surface. So what we do is look at places in the world where we have naked bedrock. Okay, that's it. You ever want to run your exposition someday? Look at places in the world that are rocks that are right age, rocks that are right type, and rocks that are exposed. There's another criterion, that in this case was accessibility. And this is where I began my first scientific expedition. These were accessible because I had just begun my first academic job at the University of Pennsylvania, the southeastern corner of the state of Pennsylvania. And they dug out a geological map. And I'm showing you here what is sort of relevant to our question. If you look at the map of Pennsylvania, what you see is everything in purple is Devonian rock. So that's lots of rocks of the right age throughout the state. Right? Well, what about that second area? Rocks of the right side. Well, one thing I would have to think it looks like in the Devonian. But Philadelphia, how do you bring this is the cartoon of what Pennsylvania looked like 365 years ago. You have highlands to the east, and then deep to the west, and a series of rivers drain from east to west. Now, if you're a county wanting to find fossils and trust the transition from life and water to life on land, this is perfect. Because you have the rays. Example, you can see rays, which are bigger than stream, even ancient something. Now, the problem is Pennsylvania doesn't have a whole lot of exposure. That third bit is just tough. So my initial expedition, if you will, became following the Pennsylvania Department of Transportation around that big old road. And we're really lucky. What they would do is they'd be digging a new road on the other place of Devonian rock. That's what you see here. This is a set of rocks about an hour north of State College, Pennsylvania. And this extends about half a mile long. It's a big red hill of rock called the hill. What we see in these layers are the strata, the ancient layers of ancient streams. Almost as soon as we hit this site in the early 90s, we started to find fossils. The first thing we started to find were two side railroad sites, big monsters like this bus. Then we started finding jaws. Here's the front end of one of these buses. These jaws would be the length of jaws. Big animal, probably about 16 feet. Then we started to find all kinds of other fish, like this little mash thing here. This is actually a mash fossil on molten fish, related to lungfish today. But then my colleague Ted Dashley, my partner in all these expeditions for the last 7 decades, had found early fish. They had left. What we had left to do an exercise software, he discovered a series of them, arm bones, leg bones from creatures that have been early. And working with the National Geographic, we Ted and I were able to reconstruct what this ancient road in Pennsylvania looked like 365 million years ago. And we have some of the first forests, an ancient stream, even middle here, that big monster fish, that 16-foot long fish. And then you get a series of other kinds of fish. We will produce all around both on land, time, and water. I realize we have about 365 million. Pretty advanced living creatures. Remember what motivated this whole class? There's a slide, it's all in practice. What are we finding intermediate? What are we finding pretty advanced living? I want something to know. Well, we get a sense of what we're after. Let's go through some of the traits. Here's on the top. It has a conical head, body of the top. Here's on the bottom. It has a flat head with eyeballs. Features that changes in the transition is the shape of the head. The other thing is if you look at creature composition, you see the head is connected to the shoulder. Here's the bone. They don't have that for the head, then independently of the body. Early live animals, early tetrapods have a neck where the head can swivel independently to look around as its leg. Finally, another big feature, given the name tetrapod or leg animal. If fish have fins, and the earliest tetrapods have limbs, the fingers, the toes, the wrists, and ankles. We're finding lots of fingers and toes, wrists, and ankles, right? We're not finding anything in the meat. But Ken and I were looking for is a flat-headed fish with fins, or there's something in that area. To get that, what we had to do was back in time about 15 million years, using analysis of other so Ted and I came up with what we were looking at, looking at places in the world that have rocks to the right age, rocks to the right height, and rocks to the exposed. We had an idea to work in Brazil. We had an idea to work in Western Europe. And everything changed one day in my office back in the late 90s. When we had a geological debate, and to settle the debate, I pulled out a college geology textbook with one grader. Now I'm at 11 of it. But anyway, so we finished the debate, and I was scanning through the book, and we ran into this figure. This is the figure that changed the direction of research for me about 15 years ago. What it is what we look for in a nutshell. The city caption that says upper Devonian sedimentary ridge, which means rock more or less of the right age. Here you see a map of North America, and superimposed on that map is the interpretation of the geology of the Devonian rocks. Now, look at the western part of North America. There are Devonian rocks mapped in ancient ocean. There, the authors identified 3 different areas, shown in red here, that had rocks that had formed in ancient rivers and streams. The first one, before that's what I just showed you, Pennsylvania. That's that ongoing Castle project that we're working on along the roadside. There's another one on top here in Greenland. That was well studied. That was discovered in the 20s, and it produced a number of early tetrapods in that bowl. Like a cartoon I've been showing you, the cartoon derived from one of the extending 1,500 kilometers east to west across the Canadian Arctic, was mapped Devonian age rock. Not that rock was perfect. It was mapped as information. So Ted and I were really excited. We just rolled around with here. We were so excited. One morning, we ran returning to the restaurant across the street, and I was. And unfortunately, he said, "Soon you'll be at the top of the tour." Yeah, seriously. We're working. And this is a colorized version of one of the sites of the paper. And it shows the stratigraphic section of here, as well as the map. And you can see superimposed over the map of the Canadian Arctic is a huge area shown in red here of Devonian rock. 1,500 kilometers. So now we have a new problem. Instead of driving our station wagons to Central Pennsylvania, we're now working several hundred miles from the North Pole where the Arctic is flying. To give you a sense, the nearest town is about 300 miles away. I'm going to show you a picture of that town, about 170 residents. There's a picture of that town. It's great. This is resupply. And as you can imagine, much of our supply is based on planes, helicopters, and so we're highly dependent on aircraft. Now, the way this affects science is that the supply chain is very long, and fossils and rocks are very heavy. That means we can't take home everything we find. So much of what we do in the field is trying to determine whether we have really good material and worth spending money. We started in 1998, at Butcher Cooking in 1998. Our first expedition was in 1999, 5 years. We started the Great Oak in the western part of the Canadian Arctic. You can see sort of what the area looks like. It's kind of flat. You can see our camp below. This is Devonian rock. So we wake up in the morning and essentially walk all over the Devonian rock, looking for the ball for the weather. We realized that the rock is still a part of it. We were in all these rocks, and there's any rock formed the nation ocean. It was pretty clear based on other people's discovery that we needed to get rocks from rivers and streams. So what that meant is, using the geological map, this is the version I showed before for Pennsylvania, it applied also to Arctic. We were in the ancient sea when it showed it. We could love to do that geologically and make moving east. It would be cool. The next year we went back and we went from east here to southern Alberta. And as soon as we did that, you see these beautiful landscapes here of mountains area underneath all red beds that contain Devonian rock formed in the earth. Took another few years. We eventually honed in on an individual valley that had good fossil preservation. And this is that valley here. What you see is in front of us, there's a big hole in the middle of the slide that shows a layer that is preserving hundreds of fossil fish that are long and fossil. So the big place, fossils, they're not very big. They're trying to find a little thing in the rock that will have those fossils inside. And what we use are the tools of geology and we work this layer. And everything changed one day in 2004 when my colleagues at the Gable and remove the rock, but we found a little notch in the middle of the slide. Look at it there. The thing I looked at, we went over that and saw we had spent multiple years or several decades. But we're kind of looking at it here, the snout fish, and not the snout of flathead. Remember I told you conical head, flathead, and big piece of tendon. What I had here in the middle of this slide, flat-headed fish, right out. Any look at all, the rest of the fish. So we remove this thing, and they come home at the base of the helicopter, so that's the first year granted scale, and it comes back several hundred miles to the 5 days there. And as we removed the first specimen, we found several more. So at the end of the 2014, multiple specimens of this flat. Really exciting. And here, removing the rock grain by grain to expose the bottom. This is what these specimens look like several months after preparation. You see the emergence of my head by see the tool that's used here. That's very cool. Several more months. Flat-headed animal body on top. And the shoulder was separated from the rest of the head. Remember what began the quest? Record to what's all the slide, fish on top, fish on the bottom. We wanted a flat-headed fish. After several decades, using tools of evolutionary biology, using tools of stratigraphy, using tools of geology. This is a flat-headed fish. This is seed specimen. Flat-head, eyes on top, has a neck, see the head separated from the shoulder, has fins with thin rays. It scales it back. Like a fish, it has scale, fin, like I already went now, has a flat head, flat top, has a neck. And indeed, when we crack open the fin, what do we find? Bones that correspond to upper arm, forearm, and butt. This is a real mixed characteristic. I'm in the right part of the right. The fish are called teutonic, yeah. And you can see here what they're salient about it. Has a mix of tetrapod fish characteristics. Like a lobefin fish, it has fins, scale, and curved jaw. Primarily a tetrapod has a neck, a wrist, flathead, and but here's a creature with lungs and gills. Here's a creature with wings that have wing bones in them, but also fin bones as well, fin rays. A real When we put it in the phylogenetic analysis, the evolutionary tree, right, right in the middle here, it really shows a fine sequence of the emergence of tetrapod characteristics throughout the bone. So what's the take-home message here? Take-home message here is to predict likely and unlikely places. We can, doing that, learn about the great transitions in history of life, in particular this one, transition from life and water to life. But the story doesn't stop there. Why? Because we can trace the bones that creature like arm bone, the upper arm bone, the human, like the top. We can trace it all the way to other animals. This story of the origin of the net. This is what? This is embedded in our body. Shake your hand. Every time you bend your wrist, you say, thank you, technology. It's evolutionary time. They live in the Northern Street, 375.
Okay. And that is us for